Hi everyone, I am Dr. C.S. Kulkarni, MD, and I am going to teach medical biochemistry. Before I start the topic, please like, share, and subscribe my channel. In the last session, we ended up with clinical applications of carbohydrates. In today's session, we will start with absorption of glucose, which includes glucose transporter 2 and 4 and sodium dependent glucose transporter. Then we have absorption of other amino acids, in which glucose and galactose transporter is an energy dependent process, whereas other monosaccharides are transported by carrier mediated facilitated transport. Then we have clinical importance of glucose, where normal fasting plasma glucose is 72 to 110 mg per deciliter, whereas normal postprandial glucose is 150 mg per deciliter. Then we have glycolysis, in which glycolysis is explained and along with a diagram. Good morning, everyone. Let us begin the topic. Absorption of glucose. The same intestinal epithelial cells have a different transport mechanism on the membrane facing capillaries. Intestinal cells release glucose into bloodstream by the carrier mechanism called glucose transporter type 2, that is GLUT2. In this diagram, we can see glucose absorption to a cell to where glucose bind to a glucose transporter on the normal surface, external surface. Once it is fixed, the complex changes configuration. This leads to closure of the first binding site, and at the same time, the binding site is now exposed to the inner side, and glucose is released. This transporter is not dependent on sodium. It is uniform facilitated diffusion system. Glucose binds to the transporter on the one side of the membrane. When fixed, the complex changes configuration. This leads to the closure of the first binding site. At the same time, the binding site is now exposed to the inner side of the membrane, releasing the glucose. The process is called ping pong mechanism. GLUT2, that is facilitated transport, is involved in absorption of glucose from bloodstream to the cells. GLUT2 is present in intestinal epithelial cells, liver cells, beta cells of pancreas and kidney. Since GLUT2 has a high km for glucose, its presence in beta cells is ideally suited for sensing high glucose level and releasing insulin. So this mechanism enables the pancreas to monitor the glucose level and adjust the rate of insulin secretion. In this diagram, we can see sodium and glucose are attached to the sodium dependent glucose transporter. This pushes glucose to the inner side of the cell. Then it passes to the basal surface of the cell. Here glucose transporter is present, through which glucose enters the bloodstream. Glucose transporter 4. GLUT4 is the major glucose transporter in skeletal muscle and adipose tissue. GLUT4 is under the control of insulin, but no other glucose transporters are under control of insulin. Clinical application. Insulin induces the movement of the intracellular GLUT4 molecules to, to the cell surface and thus increase the glucose uptake. In type 2 diabetes mellitus, membrane GLUT4 is reduced, leading to insulin resistance in muscles and fat cells. In diabetes, entry of glucose into muscle is only half of the normal cells. Coming to absorption of other monosaccharides, glucose and galactose are absorbed by same transporter, that is S-GLUT. It is an energy dependent process against the concentration gradient, and therefore absorption is almost complete from the intestine, whereas other monosaccharides are absorbed by carrier-mediated facilitated transport. Therefore, absorption is not complete and the remaining molecules in the intestine will be fermented by bacteria. Coming to clinical importance of glucose. Glucose is the preferred source of energy for most of the body tissues. Brain cells derive the energy mainly from glucose. When glucose metabolism is deranged, life-threatening conditions may occur. The minimum amount of glucose is always required for normal functioning. Coming to normal fasting plasma glucose level is 70 to 110 milligram per deciliter, where after a heavy carbohydrate meal it rises, but in a normal person this level is below 150 milligram per deciliter. Coming to glycolysis or Embden-Meyerhof pathway. Definition. In the pathway of glycolysis, glucose is split into two 3-carbon pyruvate molecules under anaerobic conditions or lactate under anaerobic conditions, along with the production of a small quantity of energy. Glycolysis is derived from the Greek words glykis, which is equal to sweet, and lysis means splitting. In this diagram, we can see the glycolysis. Glucose is converted to glucose 6-phosphate in the presence of enzyme hexokinase and conversion of ATP to ADP. Then glucose 6-phosphate is converted to fructose 6-phosphate by the presence of enzyme fructose phosphohexose isomerase. Then fructose 6-phosphate is converted into fructose 1,6-bisphosphate in the presence of enzyme phosphofructokinase and ATP to ADP. Then there is fructose 1,6-bisphosphate is converted into glyceraldehyde 3-phosphate plus DHAP, that is dihydroxyacetone phosphate, by the enzyme aldolase. Then we have glyceraldehyde 3-phosphate converted to 1,3-bisphosphoglycerate with the presence of enzyme glyceraldehyde 3-phosphodehydrogenase and conversion of NAD plus to NADH plus H plus. Then we have 1,3-bisphosphoglycerate converted to 3-phosphoglycerate in the presence of enzyme 1,3-bisphosphoglycerate kinase and a conversion of ADP to ADP. Then we have 3-phosphoglycerate converted to 2-phosphoglycerate in the presence of enzyme phosphoglyceromutase. Then 2-phosphoglycerate is converted into phosphoenol pyruvate with the presence of enzyme enolase and magnesium. This phosphoenol pyruvate is converted into pyruvate by enzyme pyruvate kinase and conversion of ADP to ATP. Then we have pyruvate converted to lactate in the presence of enzyme lactate dehydrogenase and the conversion of NADH plus H plus to NAD plus. One more important point is that the steps of glycolysis are taken under cytoplasm. Coming to the significance of glycolysis pathway. It is the only pathway that is taking place in all cells of the body. Glycolysis is the only source of energy in erythrocytes. In strenuous exercise, when muscle tissue lacks enough oxygen, anaerobic glycolysis forms the major source of energy for muscles. The glycolytic pathway may be considered as preliminary step before complete oxidation. The glycolytic pathway provides carbon skeletons for synthesis of non-essential amino acids as well as glycerol part of fats. Most of the reactions of the glycolytic pathway are reversible which are also used for gluconeogenesis. Coming to glucose entry into cells. Glucose transporter 4 or GLUT4 transports glucose from the extracellular fluid to muscle cells and adipose tissue. This transglocase is under influence of insulin. In diabetes mellitus, insulin deficiency hinders the entry of glucose to the peripheral cells. In this diagram, we can see glucose converted to glucose 6-phosphate in the presence of enzyme hexokinase and conversion of ADP to ATP and magnesium. Coming to steps of glycolytic pathway. Step 1 of glycolysis. Glucose is phosphorated to fructose 6-phosphate. The enzyme is hexokinase or HK, which splits the ATP to ADP, and the PI is added onto the glucose. The energy released by the glycolysis of ATP is utilized for the forward reaction. And hexokinase is a key glycolytic enzyme. Hexokinase catalyzes a regulatory step in the glycolysis that is irreversible. But this irreversibility is circumvented to another enzyme, glucose 6-phosphatase. Hexokinase and glucokinase may be considered as isoenzymes. Glucokinase is under the influence of insulin, but hexokinase is not. The phosphorylation of glucose traps it within the cells. Once phosphorylated, glucose 6-phosphate is trapped within the cell and has to be metabolized. In this diagram, we can see the fate of glucose. Glucose is converted into glucose 6-phosphate, and once this glucose 6-phosphate is trapped inside the cell, it is converted to glycolysis to pyruvate or glucose or glycogen or HMP shunt pathway. In this table, we can see hexokinase versus glucokinase. Regarding occurrence, it is in all tissues regarding hexokinase, whereas it is present only in liver in case of glucokinase. Regarding Km value, it is 10 raised to minus 2 millimole per liter in hexokinase, whereas it is 20 millimole per liter in glucokinase. Regarding affinity to substrate, it is high in hexokinase, whereas it is low in glucokinase. Regarding specificity, it acts on glucose, fructose and mannose in case of hexokinase, whereas it acts only on glucose in case of glucokinase. Regarding induction, it is not induced in case of hexokinase, whereas it is induced by insulin and glucose in case of glucokinase. Regarding function, even when blood sugar level is low, glucose is utilized by the body cells in case of hexokinase, whereas it acts only when blood glucose level is more than 100 mg per deciliter. Then glucose is taken up by the liver cells or for glycogen synthesis in case of glucokinase. Step 2 of glycolysis. Glucose 6-phosphate is isomerized to fructose 6-phosphate by phosphohexoisomerase. This is readily reversible. The isomerization of aldose to ketose involves the opening of glucopyranose ring of glucose 6-phosphate to a linear structure. Then it changes to the furanose ring structure of fructose 6-phosphate. Step 3 of glycolysis. Fructose 6-phosphate is further phosphorylated to fructose 1,6-bisphosphate. The enzyme is phosphofructokinase. Phosphofructokinase is allosteric inducible regulatory enzyme. This is again an activation process, the energy being derived by hydrolysis of yet another molecule of ATP. This irreversible step is the rate limiting reaction in glycolysis. However, during gluconeogenesis, this step is circumvented by fructose 1,6 bisphosphatase. The steps 1, 2, and 3 together are called preparatory phase. Here ends our session. Kindly like, share, and subscribe the channel. Thank you.